Hello, everyone. Good afternoon. Thanks to Thermo Fisher for this great invitation today at this webinar, and just for giving me the opportunity to talk about challenges concerning NGS testing in routine clinical practice, but also to share with you the main results of a study that we did recently in Europe for evaluating the performance of the Oncomine DX Express test, thanks to a great collaborative work with my colleagues from different European countries. So here is my disclosures. Here is this disclaimer. So in introduction, I will say that the main objectives of this study, we wanted to evaluate the Oncomine DX Express test in detecting different variants from FFPE samples across 6 external clinical laboratory sites selected in Europe. Therefore, a total of 6 European clinical laboratories from Basel, Nice, Naples, Rome, and Valencia and Porto contributed to this study. Initially, each clinical laboratory profiled the same pre-characterized set of clinical samples provided by Thermo Fisher Scientific. Then, in the second round, each of these clinical laboratories selected a set of pre-characterized clinical samples from their own internal biobanks, samples known to have relevant genomic biomarkers, notably for predictive response to targeted therapy. And finally, concordance was calculated between the pre-characterization data obtained on site and the results obtained with ODXET. So again, this slide shows additional details and information regarding the 6 sites in Europe that participate to this multicentric evaluation. So here is the content of the panel that we use. So the Oncomine DX Express test contains 46 genes associated with targeted oncology treatments approved or in development for different solid tumors, such as non-small cell lung cancer, colon, ovarian, and gastric carcinoma, as well as melanoma. So this IVD test can detect many deletion, insertions, substitutions, but also copy number alterations and many fusion and splicing variants on the current genes of strong interest for personalized medicine. So as you can see here, the ODX is a pan-cancer test allowing to target all the deletions, insertions, substitutions, copy number alteration, and fusions having a strong clinical relevance currently according to different international guidelines in Europe, but also in North of America and of course in Asia. So briefly, here is the ODX workflow details. Therefore, a great automation and simplification of the different steps, knowing that both library preparation and sequencing steps are realized in the same device, saving a lot of work time for the staff. So globally, on average, doing library, templating, sequencing, and analysis of 6 samples and controls takes around 19 hours in our hands. So the 82 FFP samples in total from a variety of solid tumors types were included in these ring studies. A majority, of course, were non-small cell lung cancer, followed by colon and breast carcinoma, melanoma, and many other solid tumors. Additionally, there was a wide variety tumor cellularity selected to ensure that the Oncomine panel can be used across a range of samples types in daily practice. So as you can see here on the right graph, the range of cellularity is shown with an overall range from 5% to 100% cellularity, but with a majority of samples between 45 to 100%, as determined by the on-site pathologist assessment, of course. So for this multi-center evaluation, there was a strong effort made by each of the participating sites to select samples that represent clinically relevant biomarkers that users are likely to experience with this panel test. So collectively, according to the last ESMO guidelines for NGS use that was published by 2020, all SCAT tier 1A, 1B, and 1C variants for non-small cell lung cancer were covered. Also, in addition to some tier 2 and 3 targets for non-small cell lung cancer. In another indication, clinically relevant variants in breast, colon, and gastric carcinoma were also included. Overall, the samples and variants representation in this evaluation allows for assessment of biomarker results that are likely to be used by physicians to inform treatment decision in routine clinical practice. Therefore, for the first study, each clinical laboratory provides the same pre-characterized set of clinical samples provided here by the Research and Development Department of Thermo Fisher, mainly, of course, non-small cell lung cancer, but also bladder and small intestine tumor samples having different genomic alteration of strong interest in routine practice. So as you can see here in these tables, consistent results were obtained between internal and external testing for all these samples, not only for the DNA variants, but also for the RNA variants. So now, this is a graph showing the number of gene mutations that was evaluated, which includes both CNV and NDEL variant types. The most prevalent genes represented in this evaluation, of course, was EGFR, which include the two more common driver mutation, but also the T1790M resistant mutation and different insertion in exon 20. Other clinically relevant mutations across KRAS, TP53, BRAF, METKIT, and other genes were also represented in this study. Please note that these samples were pre-characterized and they were purposefully selected to enrich for certain biomarkers. So the prevalence of the genes and variants did not represent that would be normally observed in the general population, of course. Concerning now in this study, there was a percentage of the different allele frequency in the analyzed solid tumor samples. You can see here that a large range was noted from 5 to more than 86%, but all the 6 molecular pathology laboratories, of course, include different level of allele frequency, as shown here. And this was purposefully done to demonstrate the effectiveness of the ODX to assess mutation that might exist at varying levels of alternate versus reference alleles. So this now plot represents an analysis that was performed to demonstrate the correlation between the allele frequency measurement observed with the ODX versus the expected allele frequency measurement observed with ODX. And this measurement was based on the pre-characterization method, which include various, of course, Oncomine assay and other assay. Each dot represents 1 to 49 total mutation evaluated, where the linear line represents perfect correlation between the observed versus expected allele frequency. And as you can see here, there was a very strong correlation, which provides a very high confidence that ODX is measuring mutation at the same level as previous tests that are already been validated and implemented at each site. So now switching over the copy number amplification, different cases were included in this evaluation across EGFR, MET, and HER2. A correlation between the observed and expected copy numbers was conducted to understand performance versus prior methods, including, again, Oncomine assays and FISH analysis. And as you can see here, we got a very strong correlation. Interestingly, some samples were measured at relatively low end of the amplification range for both observed and expected, which is strong evidence that ODX can be used to detect a wide range of copy number amplification in daily practice. Finally, of course, we can look here in the different fusion and splice variants that were included in this evaluation. So in total, there were 23 unique fusion isoforms detected in these FFPE samples, where, of course, a majority were red fusion, followed by ALK and TRAC, ECR1 and ROS1 isoforms. And moreover, there was a total of 6 splice variants, where most were MET exon 14 skipping and one RV7 variant. And as you can see here, all these results were previously generated using Oncomine assay, Archer, and fish analysis. So something pivotal is certainly the minimal amount of extracted nucleic acids which can be used for getting NGS results in daily practice, knowing that more and more samples are very small size nowadays, notably in thoracic oncology. So an average of 10 nanograms of nucleic acid, sometimes less, is a reasonable amount for running the ODX. And in our experience, we have more than 90% sample success rate. So it's a very high success rate using this process. So each run in this evaluation was set up in the following configuration. 6 sites selected FFPE samples, plus a positive control and a non-template control, which are included in the ODX kit. So these samples and controls were then run on 2 lanes, or half of a GX5DX chip using the Genexus integrating sequencer and associated consumables. As you can see here, the average of the total run times are in all sites was around a little bit more of 18 hours. So briefly, these slides show example batching to report between 30 to 60 FFPE samples per week for one system with a different runtimes estimation. In our daily practice, we are running 2 series per week in general with a turnaround time of 2 or 3 days for getting reports and of 18 hours from nucleic acid to reports. So the key takeaways of this presentation and concerning this evaluation now. I think first, ODX is an automated solution that can be used to simultaneously detect the different variant types and that are clinically relevant in cancer, including, of course, CNVs, indels, CNVs, and fusions. Second, ODX results are reproducible across our 6 different clinical laboratories participant in this ring study. Third, of course, ODX results using FFPE samples are strongly correlated and concordant with pre-characterization routine assay. And finally, ODX in combination with Genexus DX integrating sequencer provides testing for multiple biomarkers in around 18 hours turnaround time from nucleic acid to reports. So now to conclude this short presentation, and in order to illustrate the performance of the ODX in daily practice through a recent case that we have in our lab in Nice. This is a story of a 52-year-old and non-smoker female with no past medical history. She presented a long tumor with paratracheal lymph nodes and liver and adrenal metastases. So the diagnosis was a solid adenocarcinoma. This diagnosis was made by transthoracic biopsy. The TTF1 positive proliferation showed no PD-L1 expression and was negative for ALK. A targeted RT-PCR sequencing for EGFR was negative at this time. So it's very important to remember that. So we decided to look for different genomic alteration after nucleic acid extraction from whole tissue section rich in tumor cells. And we use the ODX and the GeneXus sequencer system. So rapidly, this is the CT and PET scan of this patient showing the different tumors before starting the treatment, as well by histological study, solid adenocarcinoma cell proliferation. Here is again the workflow of the NGS testing that we established in our lab from transthoracic biopsy until the genomic report, which was available for the physician and finally for treatment decision making. So the NGS results using the ODX were available 3 days after the transthoracic biopsy. Here is the NGS results, and we obtain an EGFR exon 20 insertion with around 12% of allele frequency and without any associated genomic alteration on the DNA and RNA NGS. So following these results, these patients received first platinum-based chemotherapy first, and after that, amivantamab therapy was administered due to the tumor progression. 7 months after starting the second line of treatment, 70% of tumor regressed, as well as disappearance of tracheal lymph node metastasis and the adrenal metastasis too. So finally, here is a PET scan 17 months after the initial diagnosis. We saw total disappearance of the pulmonary mass and of the lymph nodes met. So with that, I wish to thank all the laboratories and colleagues in Europe which participate to this great validation study. Of course, the Thermo Fisher team for all the support and my team members for doing all the experiments. And with that, thank you very much for your kind attention.